GNM Microbiology Unit 2 Methods for Study Microbes, Culture and Isolation of Microbes, Methods for Identification of Microorganisms, Microscopic Examination, helps visualize microorganisms based on shape, size, and staining properties. Light microscopy uses stains to differentiate bacteria and fungi. Electron microscopy provides high-resolution images of viruses and bacterial structures. Example, gram staining differentiates E. coli, gram-negative, from Staphylococcus aureus, gram-positive. Culture methods, microbial isolation and growth. Solid media help isolate and differentiate bacterial colonies. Liquid media support bacterial growth in suspension for further testing. Selective media inhibit unwanted bacteria while allowing target microbes to grow. Example, McConkey agar differentiates lactose fermenters, Klebsiella, from non-fermenters, Salmonella. Biochemical tests identify bacteria based on enzyme production and metabolic activity. Common tests include catalase, coagulase, and oxidase tests, used to distinguish bacterial species and their pathogenic potential. Example, catalase test differentiates Staphylococcus, positive, from Streptococcus, negative. Molecular methods, DNA/RNA-based identification. Detects microbial genetic material for accurate and rapid identification. PCR amplifies DNA to detect specific pathogens. Gene sequencing helps classify bacteria based on genetic markers. Example, PCR is used to detect SARS-CoV-2 in COVID-19 diagnosis. Serological and immunological tests. Identify infections by detecting microbial antigens or patient antibodies, used for diagnosing viral, bacterial, and fungal infections. Methods include ELISA, Western blot, and agglutination tests. Example, ELISA is used to diagnose HIV by detecting antibodies in blood. Antibiotic sensitivity testing determines bacterial resistance to antibiotics for proper treatment selection. Disk diffusion and MIC tests measure bacterial susceptibility, helps prevent antibiotic misuse and resistance development. Example, Kirby-Bauer test identifies antibiotic resistance in Staphylococcus aureus. Culture and media preparation for microbial growth. Culturing microbes is essential for diagnosis, research, and drug development. Culture media provide nutrients for microbial growth and vary based on composition and purpose. Types of culture media. Culture media support microbial growth and are classified by composition and function. Based on composition, synthetic, defined, media, chemically defined, used for research and specific microbial growth. Example, glucose salt broth supports minimal growth. Semi-synthetic media contains both natural extracts and defined components. Example, blood agar supports pathogenic bacteria. Based on function, enriched media contains additional nutrients like blood or vitamins to support fastidious bacteria. Example, chocolate agar for Neisseria species. Enrichment media promotes the growth of specific bacteria while suppressing others. Example, selenite broth for isolating Salmonella from feces. Selective media contains chemicals or antibiotics that inhibit unwanted microbes while allowing specific bacteria to grow. Example, EMB agar selects E. coli with metallic green sheen. Differential media helps distinguish bacteria based on metabolic activity or colony appearance. Example, McConkey agar differentiates gram-negative bacteria, inhibits gram-positive. Culture methods, solid and liquid. Media, microbes can be cultured on solid or liquid media depending on the purpose. Solid media contains agar, helps in bacterial isolation and differentiation. Example, nutrient agar, general bacterial growth. Liquid media, broths, lax agar, supports bacterial suspension growth. Example, thioglycolate broth supports anaerobic bacteria. Uses, solid media help isolate pure colonies, while liquid media are useful for growing large bacterial populations. Pure culture techniques. These techniques isolate a single species for accurate identification. Streak plate method. Bacteria spread on an agar plate in streaks to obtain isolated colonies. Example, used in diagnostic labs for identifying bacteria. Spread plate method. A diluted bacterial sample is spread evenly for colony counting. Example, used for food and water testing. Pour plate method. Bacteria are mixed with molten agar and poured into a petri dish. Example, used to count microbes in dairy and pharmaceuticals. Tube dilution. Method. Serial dilution of bacteria is performed in tubes with broth. Helps in reducing bacterial concentration for obtaining isolated colonies. Example, used in drug susceptibility testing. Anaerobic cultivation of bacteria. Anaerobic bacteria need oxygen-free conditions to grow. Thioglycolate broth reduces oxygen, allowing anaerobic bacteria to grow at the bottom. Example, used for Clostridium species. Gas pack anaerobic system. Sealed jar with oxygen-absorbing chemicals for anaerobic conditions. Example, used for Bacteroides in wound infections. Anaerobic jar creates an oxygen-free environment for bacterial growth. Example, used for Fusobacterium, causes dental infections. Conclusion, culturing microorganisms is vital for diagnosis and research. The choice of media depends on the microbial type and purpose. Anaerobic bacteria need special culture conditions for proper identification and treatment.